Well, I'm very excited to be here today and talk to you all and lead this panel. So a little super brief background on me, just so you know who I am. I have my PhD basically in shooting fruit flies with a laser. Done a little business development, done a little venture capital, and now I do talks at CDD. So I'm really excited about this panel specifically because we have a delightful cross-section essentially of what I think is the innovation life cycle. So at the start, at the ideation side, we've got Dr. Jon Stokes at McMaster University developing new antibodies, leading in academia. Once you have that idea, spooling it out, leading innovation in industry. We've got Dr. Josh Pottel at Molecular Forecaster, when you want to get a bunch of people together and start a company and drive a company. We've got Josh representing us there. And then as you begin to scale and expand and develop large projects, you need to bring in people like Dr. Erin Davis at X-Chem to lead those projects and implement them in ways that work and scale. So kicking us off, or actually, let's just do a quick, maybe 2-minute intro for everyone, because I can't do that well. You all know your lives better than I do.
Want me to start?
Absolutely.
I'm John. I work at Mac. I do AI for drug discovery stuff. And my lab currently focuses on antibiotic discovery because I'm a microbiologist by training. But we also have a couple projects in other therapeutic areas, antifungal drug discovery, and we're also trying to find new drugs to cure a type of brain cancer called glioblastoma. That's what I do.
I think I already did the awkward intro, so what else can I tell you about me? Something that I think will be pertinent to this discussion is I'm a big team sports fan. I talk a lot about team sports, and our team probably drives everybody crazy. But team sports is a great analogy for any kind of small entrepreneurship endeavor where people have different roles, different focuses, expertises, and that all together makes for great team building. And so I'm going to talk a little bit about that, I think, today.
Fantastic.
Hi. So what can I tell you about me? I started life off as a starving academic comp chemist. Realized about 20 years ago that all the tools out there were garbage. And so I left academia and joined industry to try and make better software tools myself. So I've had several lifetimes. I was at Chemaxon for several years before I joined X-Chem. I was at CCDC, Cambridge Crystallographic Data Center for a while. I had a 7-year stint at Schrödinger right before I joined X-Chem. So I've been here about 14 years, just trying to make drug discovery better and easier for all the other scientists instead of being that one person out there myself. So very excited to have this audience, which is a lot of people trying to do drug discovery, and hopefully we can help make it better and easier.
Absolutely. And that is a fantastic segue. I'll give you your bag of cash at the end of the talk. For the segue into the first question, which, you've all emphasized collaboration in your approaches and in your respective industries. So I'm curious if we can kick it off with a little bit of storytelling. When was a time where you had a project where the collaborative aspect was the linchpin of the project? And then also, just to add a little extra flavor on it, which collaborators does every team need?
You're asking me?
All three.
Okay, good. I would say what project isn't collaborative in the end, right? I mean, if you have the unfortunate reality of having to work with another Homo sapien, then you have to deal with collaboration and all the challenges therein. I mean, so much of what we do, in fact, is psychology, right? And so tools that lower the barrier of energy for people accessing information or using information or sharing ideas, that's enormous because humans, much like nature, are lazy. And so we all want to go to that lowest energy well. And so much of what we do from a tool provision or a partnership angle has to be making it easy. Otherwise, people just don't want to.
You can go. Okay. So I don't know. It's like when cool science happens when you take people who don't belong together and you shove them in a room and lock the door and say, okay, figure this out. You know what I mean? So you said you want a story, right?
Yeah.
All right, let me tell you a story. So I postdoced
time.
in Boston in like a microbiology lab. Technically, it's an engineering lab, but I'm not an engineer. And so a bunch of people got in a room one day. It was like a Tuesday night, and I'm like a tired, depressed postdoc, and I was like, I just want to go home. So I'm sitting in this room, and my PI was a guy named Jim Collins, and another PI who was in the room, her name is Regina Barzilai, who's like the coolest person ever. I love her to bits. We still work together. So we're sitting in this room. I'm a microbiologist. I did like chemical screening in my PhD for antibacterial molecules. That's all I knew anything about. And then there's all these people in this room from Regina's team talking about like ML and what can we do sort of at the intersection of antibiotics, AMR, antimicrobial resistance, and ML, right? And everybody just blabbing, blah, blah, blah. And anyway, the dust settled and we were like, hey, we're gonna try to leverage some of Regina's molecular property prediction algorithms to discover new antibiotics against like lab strain E. coli. Cool. I had zero hope this was gonna work. I didn't know what AI was. I didn't even think about it. It was just like magic, right? So I was like, I'll do this for a couple months to appease everybody, right? So all the professors would leave me alone. And nothing's gonna come of it, and then I can go back and do my normal work, right? So the long and short of it was I knew nothing about ML, and the folks from Regina's lab who were working on this project knew nothing about bacteria. Like, so I was asking at the start of this project, like, what are these things that you do? And they were saying, what's a cell? And I was like, oh, like we're starting from like basics, right? And I remember the first 6 months of that project went nowhere, like literally nowhere. They were trying to explain to me what they did, and I was trying to explain to them what a gene was, right? There was zero progress being made. But it was one of these things, it's like you just keep meeting like pretty much every day, like bumping heads, talking past each other, no one has a clue, and then something just starts to click, right? And then after 6 months of no progress, we had 6 months of very rapid progress because we could kind of talk each other's language a little bit. And that's, you know, when you're talking about like true interdisciplinary collaboration, like there's a huge activation energy at the start, right? Because nobody knows what anybody else is doing. And there's like this real, like everybody kind of needs to be persistent in trying to learn or at least be able to have conversations with everybody else in that room. You know what I mean? And that's something, you know, that I try to instill in the folks in my lab. So my lab, we have half of my lab is computer scientists, the other half are like wet lab experimentalists, right? And the way that they sit in the office, it's like they're all just kind of mashed together. So for example, like last year, someone who does mouse work day and night wrote an ML review. You know what I mean? And I think, you know, in the context of collaboration, just like forcing people to be really uncomfortable all the time, like kind of spawns like really interesting, you know, developments.
It's kind of creating environments where, you know, serendipitous connections and being able to actually communicate with one another kind of create something that neither party would have ever actually had in the first place, essentially.
Well, yeah, because it's like, you know, if you're like a hyper expert in, you know, protein biochemistry and you end up working with 100 protein biochemists, it's like, that's kind of cool, but you're not really like expanding necessarily your horizons as broad as they could be, right? So yeah, I think, and in drug discovery, as we all know, like it takes like a total village, you know what I mean?
Absolutely. And I think talking about villages, Josh, I imagine there's a wealth of opportunity for collaboration with you, right? As a CRO, I mean, all you do is collaborate every day. So I'm curious what if you have a story as well where that kind of collaboration drove it.
I'm not sure that I would pinpoint an individual story other than kind of the way we built the company organically. The idea of collaborating was crucial to us. I was fortunate that, I can't remember who told me this at a pretty early stage of my life, I guess, was no matter what you do, you'll always work with people. And you better figure out how to work with others because you're gonna have to. And so I did that kind of unintentionally as best I could. And then as we built the company, especially as a service provider, especially as a computational service provider, you need to work with others because you need others to actually make things. I'm all for, you know, kind of preaching what computational tools can do, but at the end of the day, a computer screen is a computer screen, and you need to actually do something in the real world. And so what we do internally and externally is collaborate. Maybe a story I can tell you is how we built, everybody, you walk into companies and they have the company values on the wall, right? And it looks a little, I don't know, preachy or, whatever. We have that too now. But I can tell you the way that we made that. And it was entirely from a third party organically. This wasn't something that was top down. It wasn't something that was said, we're going to be this way. It was a third party interviewing everybody in the team when we were just a team of 4. So everybody sounds grand, but we were 4 people. Interviewed us individually, independently, different questions, had discussions. And then from those notes, that third party synthesized kind of what our company values were. And she expressed kind of, I don't know if it's surprise or I guess it was unexpected that there was already this culture that had been built in the company. And that's the kind of ground up organic team building that you want. And collaboration was one of the things that came through, that we all work together. And today we're a team of 15 and everybody brings a different background. It's not quite as drastic as engineers or ML folks and biologists. But now we have grown to have some life science experience, software engineers. But even within just chemistry, you can get quantum chemists and then we don't have a peptide chemist, but let's say a peptide chemist. And they would also probably speak past each other for a little bit. So as we bring those people together, as we put them on projects together, we're very fortunate to have everybody in Montreal. So everybody's on site, tapping each other on the shoulder. They're not forced to be in the room. At least I can't say that publicly. But they are on site, and they do work super well together. And it makes a fun environment, which I think that's the other ingredient that makes for good research.
Absolutely. Thank you. And so we've talked about, the collaboration as kind of the underpinning. So I want to switch gears a little bit and talk about our newest collaborator, which we all know about, which is, ML AI. So I would love to hear about a story from each of you about the computational tools that you've been able to fold into your existing processes, and how you've, a story where you've seen a successful ROI in that implementation, and it's enhanced the workflow as a whole. And if you also mention model for the for the experts in the room, who I am not. Whoever wants to go first.
That is a loaded question because it turns out that ML doesn't work most of the time. And knowing where it's applicable and where it's not is probably one of the most intelligent things that a human being can do. I think we're all very cognizant of the hype. Are you familiar with the Gartner hype cycle? Have you ever seen this before?
What, no, so yes,
oh my gosh.
Please.
So it is, Gartner, the big consulting firm, they track hype cycles of technologies, and it has this big bell curve, and there's all these, heightened expectations, everyone's frantic, and then there's a steep drop-off after a couple years of the technology called the trough of disillusionment. And if you look at the hype cycle for any AI technologies over the last 8 years, as tracked by Gartner, everything peaks, and then it's like that roller coaster that kills people at the end. Everything falls into the trough of disillusionment, and most AI doesn't come back out. And it has been, and I say this as someone who has been very, very successful with ML, and it can do phenomenal things, but knowing where it's going to be very applicable, the domain of applicability, where is it going to move that needle, where is Gen AI or all the other things going to actually do something interesting, and where is it absolutely never gonna, and then kind of avoiding those in order, don't ride the roller coaster, ride the roller coaster to where it's gonna go and be very contributive. And I think every one of us probably have oodles of stories of garbage in, garbage out, but also where it's been really fantastic. And so, look, if you put 100 data points into a machine learning model, it's gonna have a bad day, right? But I think this is one of the things that wooed me over from Schrodinger into X-Chem was that the Dell data, which makes billions and billions of data points, is actually enough. But even that is still within a domain of applicability. and it cannot extrapolate into chemical space. So you have to be very careful about where you're going to use these things. And Brian, where are you? Hey, thanks for making my case for me, buddy. You know, it can be very, very valuable. And then other times it's just, man, some of the molecules that come out of Gen AI, I can use them to torture med chemists with. They're so bad. So it's careful application and staying out of that trough of disillusionment, I think.
Also, great indie band name, Trough of Disillusionment.
Oh, it's been, you should check it out. It's fabulous.
I'll just broaden the point because I couldn't agree more. But when I did a startup course at UCSF, that's my formal training into entrepreneurship. And one thing that really stuck with me and is repeated throughout kind of the entrepreneurship community is don't build solutions in search of problems, but identify the problems and then identify the correct solution. And it's the same thing here. ML, AI, I mean, call it whatever you want. We used to call it stats or QSAR for those back in the day.
QSAR? What?
Yeah, I'm not that old. But you know, these are good tools or solutions for certain problems. And jumping into Dell's to say, hey, these tools can help here, brilliant. Saying, hey, I have a target that's never been drugged before, there's no data points, let's just throw it into, you know, some generalized model that we think is gonna solve this for me and put everybody out of work, you know, is not really the right approach. At least that hasn't been successful across most of our industry.
Yeah, there's a lot of hype, like too much, I think. All right, so I'm gonna tell you, so I'm an antibiotics guy, which I don't know why. Actually, I know why. I don't know why I still stay in it though, because you can't make any money and no one wants to pay for it and yada, yada, yada. But I'm gonna paint a bit of a I'm gonna try to justify my existence, okay? Can I do that? All right.
Absolutely.
We're gonna have like a therapy session right now. All right, so this year, 1.5 million people are gonna die from drug-resistant infections, right? That they probably wouldn't have died from 20 years ago. And it's because we haven't discovered a new antibiotic since 1987. So that's a problem. But antibiotics are really interesting because I've invented the best antibiotic ever today, and it gets on the market tomorrow. Doctors are gonna put it on the top shelf of their medicine cabinet, and they're not gonna give it to patients until their patient has failed every other antimicrobial therapy and they're about to die, and then they'll give them your new drug, and then they're gonna take it for a week, and then they'll be cured, and then they go home. Right? So given that it costs $2 billion to make a drug and like a few hundred people are going to take it for a week, you're never making your money back, right? Yeah. But we need antibiotics, like full stop. So if you need something and no one wants to pay for it, what do? You have to make it cheaper, right? And then, you know, so we said maybe ML can help us get to new antibiotics faster and cheaper. Maybe it's still an experiment, we don't know. But we wanted to apply generative modeling to help us, you know, broaden the chemical space of antibioticness, if you want to call it that. And a couple years ago, we were messing around with some of these existing generative AI models. And like you said, a lot of the times the molecules that were outputted were just unsynthesizable, like they looked insane. Or if they were synthesizable, it was like 30 steps and 10 grand a MIG or something. You know what I mean? So I was like, I can't afford to do this. We need to do something else. So what we spent a lot of time working on over the past few years are like molecular generation models that are constrained to predefined synthons and synthesis reactions, right? So last year we published a paper, you know, where we developed this model called SynthMol, which uses a collection of, it's like 130,000 molecular fragments in 13 reactions, and that gives us access to a theoretical space of 30 billion chemicals, right? Fine. And we synthesized, like we trained this model to make antibiotics, we synthesized and brought them into the lab, and some worked and some didn't and whatever. But I think, you know, at a high level, what we need to do, like if we really want to apply ML for real drug discovery, like actually, is try to incorporate a lot more biological complexity up front, like during the training process. Because what I see a lot of now are folks, this is gonna get me in trouble with some people and I apologize, but there's a lot of efforts right now focused on like small molecule protein interaction prediction, right? On the heels of AlphaFold and whatever. That's great. But there have been many, many cases where we discover a nanomolar binder of a protein, and you introduce that molecule into cells and nothing works, right? And I think it's because the model is trained on such like a reductionist problem that it's difficult to predict whether that's going to work when you introduce it to a complex biological system. So something that I'm trying to drive home with my trainees anyway, they don't listen to me, but whatever, is like, you know, if you want to train a model to design a drug to cure a disease, you have to train that model in a manner that captures the biological complexity of that disease. You know what I mean? And that sucks because it means you almost by necessity have to gather your own training data. And those models end up being a little bit clunky and difficult to work with sometimes, but it is like, if you're trying to solve a problem, that's what you have to do, you know what I mean? Does that make any sense?
But John, biology is hard.
I know biology. Dude, and it's not even that biology's hard, it's like, it's not ones and zeros, right? Like biology is like very messy and there's stuff that's connected and sometimes stuff's on a little bit and a lot, and you know, it's just like, it's like so crazy.
You have to bring that. You have to bring those considerations up front.
Oh, I know.
The most perfectest small molecule in the whole widest world for one particular protein target with the bestest properties ever there were is still not necessarily a drug.
No, I know.
To exactly your point. And so I guess you know, when the world was very reductionist at how do I optimize, and I say this as a comp chemist, how do I optimize the, you know, free energy perturbation change of the specific atom with this specific protein? That's an easy way to think of the world. But then you have the rest of the owl fundamentally to draw, right? Which is the biology and the biology process. And so I think that is something that I'm very excited to see ML get more and more into, because where ML has been so useful for the research that we're doing is looking at all these other convoluted biological pathways that correlate to a specific therapeutic state compared to, you know, healthy populations. And then my little tiny human pea brain just cannot conceive of the data that it can. And so I think that is the direction that is going to be yielding so much value, because you have to capture those things up front.
Mm-hmm. And like the biology is one thing, right? But the so we all appreciate that drug discovery is like fundamentally a multi-property optimization task when you think about it. And the biology is one thing, but then there's all the annoying stuff that like only, I don't know, I don't care about, but I know is important. It's like, is it sufficiently soluble, right? Can it get to the right anatomic site at the right concentration for the right amount of time to
You have those
elicit an
stupid
effect?
livers to deal with.
Like all that crazy stuff, right? Stuff that like is tangential to the thing you're trying to fix. But like all of those other properties are where a million stumbling blocks are encountered, in my opinion, like quite late in the life cycle of a given molecule, right? So the question becomes, it's like, can we train, you know, even reasonable multi-property prediction algorithms such that all the molecules that are being outputted from the model in silico before you even go to synthesize something has all of those other what I'll call like annoying properties built into it, right? So you're not spending a ton of time and money on stuff that is inevitably going to fail due to like liver tox or whatever. You know what I mean? The issue is, you know, and we're trying to figure this out now, it's like we're fundamentally data limited across all of these different ADMET properties. You know what I mean? So it's like we're constantly living in these low data regimes, which as you know is not ideal for training ML models, right? And even if the data does exist across all these different ADMET properties, for example, it's oftentimes the chemical spaces that's explored in all these individual assays are like fundamentally incompatible. You know what I mean? So something that I've come to realize, you know, doing this stuff, it's like the best AI scientists I know are also the best data scientists I know. And it turns out they spend probably 20% of their time doing AI and the other 80% of their time like squishing data between their fingers to see like, is this reasonable data on which to train a model? As like unsexy and boring as that is, like they're never fun to talk to, but like, it's just like they're the most productive because they understand their data the best. They understand how to interpret the model outputs the best. You know what I mean? So.
On top of all the scientific challenges that you mentioned about the things that we don't necessarily want to care about, we haven't even got into the logistical challenges of doing that, right? That going for kind of the holistic shot on goal that doesn't necessarily have those interim milestones, even if they're useless, doesn't address the risk aversion for capital, doesn't address regulatory concerns. And so this all comes back to collaboration and getting everybody kind of to row in the same direction. And that's really hard, insert 4-letter word there. It's very, very difficult to get everyone aboard. There are a lot of startups, ambitious entrepreneurs that want to do these things that we're talking about, and they want to work together. But there are a lot of people that are not necessarily ready to do that. And so we try to kind of usher things along, at least in the right direction, but that's not trivial.
Absolutely. So kind of building on that point, we've talked a lot about the current models that we have access to. There's a struggle for good data. How would you recommend people to approach this problem? Because there is a lot of hype and people, are getting grants to do this work. So how do you set yourself up to do it successfully and not to fall into the pit of despair?
We're in drug discovery. We live in the pit of despair.
I
don't know of any other career that says you have a 3% chance of success, and people are like, sign me up.
It's moved up to 3. I would say, like, don't do what everybody else is doing. I mean, like, we need all kinds, right? We need the ambitious person that says, I'm going all in, all at once. You need some people that say, the data is never going to be sufficient, and I'm going to try to find another workaround to make this go. You need support people, hopefully, to keep us employed. Do things that don't scale. Try something different. Find people that support you, and work around. We don't all need to be doing the same thing to reach, one objective. I'm sure everyone in this room, I'm sure, has shared an objective with somebody else and thought of a completely different way to reach that objective. Go do that. You don't need to be like everybody else. You just need to be able to storytell your way to convince somebody that your way makes sense. It doesn't even necessarily have to be better than somebody else's. It just needs to make sense enough that you'll get that collaboration and the support around you.
I'm reminded of, in population genetics, diverse ecosystems are much more resilient. So if you have a monoculture of ideation, that's not going to be a resilient system.
What was the question again?
Essentially, how do people, when you're approaching, building an AI model, how do you?
So it's like, you shouldn't try to be using AI for everything all the time. I think that's a trap that a lot of people are falling into now. It's like there's this shiny new toy and everybody wants to use it. That being said, though, it's because a lot of investors will only fund stuff if you're using AI, which is just the dumbest thing in the world, but whatever. And I like the comment earlier. It's like, AI is a tool, right? It's a hammer. It's a fancy hammer, but it's just a hammer, right? And you can't build a house with just a hammer. You know what I mean? So it's like, if you actually want to develop a drug that does something beneficial, that's your problem statement. Focus on that, and then ask what are the sets of tools that exist, that I can get to that solution easier. And if it doesn't exist, and you're forced to invent something, then go invent something. But something I'm a huge proponent of, it's you should keep projects as simple as possible for as long as possible. You know what I mean? And if that means you don't need to use some fancy ML model to help you get there, just don't, right? Because what are you trying to prove? You know what I mean? Yes, and when I say that, all the ML people in my lab get mad at me because they're like, well, why am I here? And it's well, because sometimes you need to invent stuff because stuff that exists to solve these problems doesn't exist yet, right? That's why you're here, right? But if we don't need to do fancy hard stuff, don't.
The answer is, well, why do you want to build an AI model? I'll just totally echo what Jon said. Why? What do you want an AI model for? Because one, it's not always the answer, and two, it's not necessarily always going to work. But more importantly, nobody has on their annual objectives build 3 AI models, right? Their objectives are attain a TPP, make a drug, figure out this pathway, right? They're all trying to solve a science. So this is where that collaboration bit comes in so well. Our world is so tiny, even though it feels like it's often huge to us because we're in a little goldfish bubble. But It's got enough people doing cool things that you can find that collaborative spirit. I mean, so creativity, and this is applicable to business too, and I say this as somebody who also, works at a CRO, like, we want to solve problems, I want to solve problems, we'll do all kinds of creative things. So you want to build a model and you want data, come talk to the people who can make that data. This is not a restaurant. You don't have to order something off of a menu. Maybe I'll give you a sweet deal and we'll do a cool thing together and we can talk about it. So you're not held back by any notions other than, I think, the basic levels of physics. And beyond that, it's up to human creativity. So what are you trying to achieve? And have you explored all the various options to it? And I will take a hot moment to really nerd out for a second. Are you familiar with the Kobayashi Maru?
Remind me.
Does anybody know what the Kobayashi Maru is? Okay, so real short story, because it's very, very applicable, and I teach all my people this. The Kobayashi Maru is the test in Starfleet Academy when you are so very old.
I'm asking the questions. We're not asking that question.
I'll keep it real short. But basically, it's an unwinnable challenge, right? So before you graduate from Starfleet Academy, it's a Star Trek, they put you in a simulator and you get attacked by Romulans, and either you can save some people who are stranded in space or you can defeat the Romulans, but you're going to lose some way, and all they really want to see is how are you going to solve the problem. Well, Captain Kirk said he didn't like those rules, so he snuck in the night before and he changed the rules of the game. He reprogrammed the system. So he got in there, and I can't believe I'm doing this on a panel. And he saved the people, and they were like, oh my God, what did you do? And he said, well, I didn't like the rules of the system, so I changed them. They let him graduate, but he got beat with a cane or something. I don't remember. So I tell all my folk, that you are not constrained by rules of the system simply because this is how business is done or this is how science is done. There's fundamental hypothesis, but what assumptions are you making that you don't have to make anymore? And then go out and solve your problem. So just to sum it all back to what Jon was saying, why? What do you want to do this for?
Absolutely, and I think that's an interesting segue, kind of changing gears a little bit for the second to last set of questions. So thinking about, the rules of industry, proprietary IP takes a big precedent. So in balancing, right, open source and proprietary data, I'm curious, as we're building large data sets, as we're making ML and AI tools to digest them, where do you see that balance, and where have you seen how to navigate trade-offs there already? Whoever wants to take it first.
All right, I'll talk through the lens of an academic. I publish, right? So it's like I answer emails all day. That's why I was late getting onto the stage. I was ripping through a bunch of emails that I forgot to get to. So all of the data, training data is open source when we publish. All the model parameters are open source. All the model checkpoints are open source. You can run the models on your own data. Everything's open source, right? The one thing that we do get IP on before we publish is the chemical matter, right? Because we want to eventually turn those into a drug. And in my opinion, the value is held in the molecules themselves. All the other stuff is just the methods through which you got there. And I'm a huge proponent of all of those methods being freely available to everybody who wants to use them. I understand data is often expensive and tedious to acquire, and that's why some folks, rightfully so, say I don't want to show this right now. But when it comes to models, and this is where again I might ruffle some feathers here. If you think you have the best model today, you might, but wait 6 months, and you won't, right? So people trying to get hyper protective about their models and say I'm not going to tell anybody how this works. It's that it's not cool, but let's have another chat this time next year, and who's going to have the best model now? It's not going to be you, right? And I think it just benefits everybody, across domains, specifically in the context of ML model architectures and stuff. It's just let everybody have at it, and then everybody can leverage those architectures for their own drug discovery tasks, right? But I mean, that might be against what you think, so I'm sorry. I don't know.
I mean, it doesn't have to be, right? I think the status quo industry perspective is, well, no, we have to keep everything secret and no, it all belongs to us. And that's how we're going to rule the world. You quickly realize that that's a farce, number one. Number two, you can change the rules. And you can decide that, okay, with this system in place and these are the constraints and the parameters within which I need to play legally, I can do things differently and I can do things my way. Here I can share a quick anecdote. Has everyone heard of these cache challenges from the Structural Genomics Consortium? Some nodding. Okay, so this is an open source competition, and I do this because I hate that it's a competition. This should be a way to help the community. Where basically the SGC and now another organization called Conscience Medicines, which spun out from other things, which I think X-Chem actually supported from very early days, as did we, to bring computationalists to make predictions on a interesting, important target in drug discovery, and then they will test those compounds in the real world, in the lab, and all the data becomes public. They also encourage everybody to publish their methods and share the way that they approach the problem, all with the goal of advancing the field forward. Now, the reason that I think the word competition sucks is because that pits people against each other, and they say, well, I need to be better than the next person, and no, I'm not going to share what I did because that's going to enable this other person to do what I want. And so ultimately you end up with I don't know what the numbers are today, but for the most part, it was about 70 to 80% academic participation and not industry. Now, when the first one started, I said we supported this from very early days. We had a lot of debate internally. We were a younger startup then. We were a team of still 4 or 5 at that point, debating whether this was a risk for our company. Now, I would tell you that there is not much business upside for a startup to take this risk. You can really only fail. If you have case studies and you have examples of your technology working, you can kind of only be made to look the fool if you turn up nothing. And so from that standpoint, you would say, well, industry shouldn't participate, and it's a big reputation risk. Research is hard, things fail, and at the end of the day, you get one number out, one data point, you didn't find the molecules, you lost the competition, you suck. We said, well, let's throw out the competition, let's change the rules, and let's call this a learning experience for us and for others. And we'll use this as an opportunity to test new methods, to try new things. We'll publish those things. We're happy to put all the data out. In addition, we had the cherry on top that we could use this now to train people in our team on lower risk projects. Side note, total respect for the people in the wet lab that actually have to spend the time testing those compounds. I don't take that lightly. My postdoc, we were forced to spend time in the lab to see how much time it takes someone to test that one compound that you looked at a data point and you said, yeah, that one. And then they spend 3 weeks to 3 months either making that compound or testing that compound and doing all the data analysis. But we changed the rules to say, we're not going to focus on this competition stuff, and we'll control that narrative, and we'll push that data out. No problem, we'll publish our methods. We're not publishing our source code. I mean, we still need to keep something that remains within the company. But all those learnings, we're even doing postmortems after the studies are done, which is where, in my opinion, the real work begins. If you're really learning, you need to go back. You don't just say, oh, it worked, oh, it didn't work. You need to go back and understand why and make changes. And we've made changes within our company because of those things. So the more you are curious, the more you keep an open mind, you'll see that you can find benefits to a lot of different models, and it doesn't always have to be that everything is siloed and secretive.
Erin?
Remind me of what remind me of the question, because I could totally riff on what's being said here, but.
Absolutely. So it's the balance between open source and keeping everything proprietary, and where you've had to navigate that.
That is a precarious question, very, very precarious, because in a utopian society, everything would be open source, because at the end of the day, we're scientists, right? We want to learn, we want quality data, and data is very hard to make a quality statement on. Just anyone who's spent any time in Kemble will tell you that, right? but data is expensive to generate. And we still live in a capitalist society, and I live in the United States, unfortunately. And so, you know, the funding there has just gone up in a giant dumpster fire. And this makes it an even bigger question. So I worked with industry for the bulk of my life, including you know, all of the big pharma. And human-based operations are expensive. Generating that data is expensive. I did a couple years of the bench as well. That's expensive, even if you're an underpaid, you know, postdoc slave. So you're in this weird position of trying to be altruistic for the betterment of society, but you also still have to pay your people. And lab space is expensive, and reagents are expensive, and health coverage and co-pays in America is expensive. And so, you know, there has to be this counterbalance. And how do you do that while people are dying of diseases, right? I mean, it's really precarious. And I think we're at a really interesting time now with AI gobbling up everything. And these old models that have held for so long are now like creaking at the knees. And this is particularly relevant to me because I just had a rather spicy call with a publisher, you know, and we're trying to re-up our subscription again, and these are the keepers of this data who don't pay the people who actually publish it. And one of my favorite memories of Alex Clark was giving a talk at ACS. It was just in fuego, in the faces of publishers talking about the gatekeeping of this incredibly valuable data as like as a corporate thing. But they have humans to pay as well. So there's no good answer, and I think you have to do what you can within the context of survival and research, because this stuff still is pretty expensive.
Absolutely, that makes a lot of sense. So one more quick question here to kind of cap us off before we open it up for some Q&A. Looking to the future, you know, we've kind of talked about some stories of what's happened up to now. So to all three of you, what are you most excited about either an emerging tech, something you're evaluating, a model you're building, or tools you're implementing that are going to land at the end of 2025, early 2026? So what do we have to look forward to? Whoever wants it first.
In 6 months?
Or next year.
Or next year? I need to think about this.
Josh, do you have an answer?
You're not going to have the new best model?
I don't have the new best anything ever. Actually, I kind of know what I'm interested. So, all right. So remember I said Regina Barzilai's name like, 20 minutes ago? So one of her students, a guy named Gabriel Corso, super cool guy. He he's not her student anymore. He defended like a month ago. He released something called Boltz 2. Did anybody see this Boltz 2 release? Yeah, and it was like awesome. So I think that's for those of you who don't know, it's a foundation model that predicts, you know, whether a small molecule will bind to some protein, and built into this is an affinity prediction algorithm as well. So supposedly it will tell you how tightly some molecule binds. There's a ton of hype like around this, which, you know, earned for sure, like realistic in the real world, unlikely. But I think it's a step in the right direction, right? So if you can have these foundation models that can predict small molecule binding to proteins, right? And you couple that with, you know, a model that only outputs synthetically tractable molecules, you know, R synthmol or pick and whatever, I don't know, I'm sure there's a bunch now, you know. And then on top of that, you know, foundation ligand protein interaction model, you can start to layer in a bunch of these other, you know, I'll say like, you know, more like clinically facing models. And when I say clinically facing models, I mean things like, is it likely to work in a whole cell? Is it going to be toxic? Yada, yada, yada. And I think now you're in this sort of realm where in silico, you can design a molecule that you know you can make, ideally for cheap. It sticks to the thing you kind of want it to stick to. It's likely to have all these other physicochemical properties. You know, you're in a position now where you can be fairly confident when you start to synthesize stuff and bring it into the lab, where, like you said, it takes a ton of time and costs a ton of money, right? where the probability of success is going to be like measurably higher than if we were to do it, I'll say like through an unguided mechanism. I think those pieces, like all of these pieces are like kind of beginning to exist now. And it's just a matter of, folks in the field starting to put them together, right? Into what I'll call, for lack of a better term, something that's like sort of like end to end, at least preclinically, right? Something where you can design a molecule. You can already start purifying your protein because you're fairly confident it's going to stick to your protein of interest. It's probably going to work in a cell, yada, yada, yada. And I can see that happening over, 12 to 18 months.
Well, I got an easy answer for you about what I'm super pumped about in terms of new technology, and that is something that I've been working on for about the last 9 to 12 months at X-Chem, and that is fundamentally taking DNA-encoded libraries and changing the paradigm from hit finding to protein mapping. So if you have screen data that is very, very high quality, very high signal to noise, now you are basically taking 300 billion molecules and probing the protein with it. So we're turning these into structure function maps that, as one of my comp chemists like to say, the protein knows what the protein likes, and it does not care what you like. And so we can take these one experiment now and fundamentally kind of map out all the different atomic spaces that the protein has demonstrated interest of. And when you think about the way a DEL experiment can be run, it's not just one protein. You can look at individual point mutants, high concentration, low concentrations against challenge inhibitors, against known drugs, all in one single experiment. Now, this is a metric butt ton of data, and the challenge has been figuring out how to turn that into these structure function maps, especially across different potential modes of action, because there's a number of different biologies we can explore at the same time. And so turning this into a mapping technology has been super, super exciting. And for me as a scientist, the reason I'm so excited about this is because it's basically front loading a program. If I can give you, years and years and years worth of SAR, instead of going through these expensive DMTA cycles, but we go through one gigantic experiment up front, now, in theory, you've shortened the time scale, and you already know what your protein likes in response to a very diversely, specifically designed sets of libraries. The information is redonkulous, and that's a technical term. But it's the same data that goes into the ML that's worked well. So, I'm excited we have some early stage partners who are using this in a prospective fashion. The enrichments are shocking. Like, I don't actually believe them at first. When we look at retrospective. So we'll be talking more about that in the early fall, but I'm super fucking pumped.
That's also a technical term.
Totally.
That's going in the publication.
That's a title.
These people are much smarter than me on the technology, so I'll step aside from that and ask, how many students do we have in the room? Hands up. No students.
There's a couple.
Two.
There's a couple.
Two. All right. No, okay, cool. You shouldn't be worried. How many Canadians do we have in the room? Okay, that's a vast majority. Sorry.
It's okay. We suck.
I'm a dual citizen, so
Okay.
I regret that high five. I'm excited for kind of continuing this rallying of Canadian biotech. I think that about, 4 or 5 years ago, we had really hit a stride. The community had a couple of company exits, some good, some less good recently. There's been an influx of people. Unfortunately, markets are not doing great, and so capital is harder to come by. But we can't kind of stop that momentum, and we need to keep going from kind of ground up, building new efforts, building new companies with these technologies, with collaborating. It's something that Canadians do very, very well, is working together, having spent time in some other places around the world. I don't want to make it sound like I'm this world traveler, but we do this very, very well. It is our competitive advantage, and the more that you students and you Canadians keep pushing that ball forward, we can rebound from this trough. It might not be. I hope it's not disillusionment, but we're certainly kind of trending a little bit in the wrong direction now, and it's time to push against that and follow those bold ideas, try new things, and that's up to all of you, all of us, and I'm cautiously optimistic that we can figure out a way to do that together.
What a fantastic way to end the panel on such a hopeful note. So please join me in thanking our fantastic panelists. And now we have a little bit of time for Q&A. If anyone has any questions, we'll have some runners. We've got a question over there.
Awesome. Yeah, hi guys. Yeah, great talk. Ralphie, I'm on the product team here. I think this is probably just like another more broad question, but we've obviously jumped on the AI plus bandwagon, helps companies get funding, right? It helps users have access to tools that they wouldn't otherwise have. Something that I'm always thinking about and things that I remember running into when I was on the support team is, advice for companies who they're collecting all of this data. They're not really sure like how they should be curating the data in their vault, for extraction later on, whether it's developing their own models, passing it into their own models, or passing it into some new stuff we're developing. So just some advice there. Maybe me thinking forward of like whether it's a service we offer to help consult these companies on like how to store the data, or if it's just an addition to our AI offerings where there's like a more curated way to capture metadata and annotate this information. Just riff off of that.
There's a whole world of ontology providers for a reason. You have to remember my foundational hypothesis was that humans are lazy. And so that ensuring that all of the metadata, especially the things that are a couple nodes away, is often not what a busy chemist or registrar wants to do. So the more that you can look at ensuring some kind of commonality across the taxonomies for metadata, the more interested I am, for sure. I know that is definitely something that holds it all up, because a machine doesn't know that CHO and Chinese hamster ovary are the same, right? It has to be told, this is an apple, this is an apple, this is an apple, compare them across the board.
Yeah, and we have we have ontologies built in as well. So, Alex Clark has been a huge help in that. So maybe it's more like educating new users, like maybe forcing that upon like people who are interested in the AI ML space, or not even, right? It's like if you want to set up your vault in the perfect way, right? It's like, here are your ontologies, set that as like a forced, essentially, field to annotate all of your assays. Yeah, I like that.
Yeah.
You can also be critical. I'm here to take feedback, so yeah, curious.
I'll speak from like experience. Like when data sets get like too complex and there's like layers of stuff, it just sucks to work with, right? Like if you're trying to, like, simplest case, it's like I want to train like a supervised algorithm like on a binary classification task, right? Here's a whole bunch of molecules, yes, no, right? That's the task. When you I hate looking at data sets where it's like, it's not, everything's in the same format and you have to go to like layers and layers of some data bank to get the relevant, nobody wants to deal with that. You know what I mean? So I understand that, metadata and whatever are, I'll call them, what's a good word for it? Necessary evils, let's say. But like from the perspective of like model training, it's like you want everything that you need and nothing else, just like on one sheet, like one CSV file. You know what I mean?
Yep.
And as boring as that sounds, like that's the most effective data to work with, right? Or at least when it comes to like, storing that data, and then you can take that data to train something with it. You know what I mean? It's just like, sure, if you need all that complexity, like put it somewhere, but don't make it so like I have to navigate that complexity in order to train my model. You know what I mean? When that complexity isn't going to be important for my model to run predictions or do generations or whatever. You know what I mean?
You're going to like something I'm going to present on later, I think.
Okay.
So.
Fantastic. Yeah, we've got a question over here.
First of all, thank you very much for the talk. It was very interesting. I just graduated from my PhD, so Simple question. Thank you.
Congrats.
I guess my question would be, I'm fresh from academia, and I'm a biologist, so I don't have like much background in like computer science or data analysis and stuff like that. Data science, I guess. So what would be like your recommendation for someone who's trying to transition into this industry, and like what kind of skill set you're looking for?
All right, I can answer this because I'm a microbiologist, right? Like I spent, I don't know, 6 years like doing this all day, right? And then I found myself in this postdoc with these computer scientists who were talking gibberish, and I was like, all right, I guess I have to do this now, right? They're like, to get good at do you like play sports or anything?
Yeah, like a lot.
All
of
right,
different
like what sport do you play, for example?
Sometimes soccer. I don't know, like a lot of
Right,
different
okay,
things,
but to get good at soccer, you can't like dabble in it, right? It's not something you can do once a month and be like, I'll figure this out. Like you have to kind of like head down if you actually want to be a good soccer player, right? If you actually want to be a good anything, it's just like you have to invest the required amount of time and energy in order to get good at something. So how I ended up doing it is I had this problem that I had to solve. I wanted to discover a new antibiotic to treat lab strain E. coli. Who gives a shit? But it was a good test case, right? And then you just don't give yourself an out and you just figure it out. I can tell you there are enough freely available online resources for ML and chemistry that you can learn this by yourself, right? You have a PhD, you're smart, right? You're way smarter than I am. I'm the dumbest person in this room by like a country mile. Right? And if I can figure this stuff out, you can too. I promise you. Like if you actually head down, start playing with whatever data sets you're interested in the context of whatever disease you're interested in, right? And you want to leverage ML across those two things, the data and the disease. In 6 months, you'll have this. I promise you, right? It's going to be a shitty 6 months, but you'll get there, right?
It's all right.
But
I'll take it.
you will. Like before it gets cold outside, you can be doing this.
Thank you.
I'll quickly ask you one question, which is, what do you want to be doing? And it's a rhetorical one because you probably don't have the answer. You might know what you don't want to be doing, which is just as helpful. The only way to find out what you do want to do is by trying things, to Jon's point. And so if you want to be doing computational biology using your training, then that's one way to go about it. I would say, speaking from a position of privilege on the other side of the table, I never applied for a job. I just walked into my own. You can find a place for what you want to do and for your expertise. It's about, I hate to be cliche, but it's about networking, meeting the right people, finding that right connection where you fit within that team. Your PhD in biology and your knowledge and your experience as an individual, I guarantee you will be useful somewhere. It's a question of, what do you want to do? What gets you up in the morning? What gives you that energy? That doesn't have to be something that you're good at today. But it can be something you're passionate about and you're excited about. And that will get you through those rough 6 months that Jon is talking about.
Wise words. All right, so that is time. We did it, y'all. So one more round of applause for our speakers. Round it off.
Great job, everybody. Second time I've heard Jon speak, and he's the smartest guy I know that keeps trying to convince you he's dumb. But I like, he said forcing discomfort, which I think is hugely important. And to your point about career, put yourself in uncomfortable places. And my staff tell me I make them uncomfortable all the time, so I think I'm doing my job. Speaking of discomfort, getting to lunch is a little bit difficult in that there's a couple of choke points, so follow the CDD people. Give yourself a lot of time before coming back because there's a couple of elevators where it takes a minute to shuttle everybody. But we come back here at 1:30, but right now you get lunch for being such a great audience, so thank you very much. And now I move to the slide.